where we are going to be looking at the topic on proteins. Proteins are a very important component of the human body. They are one of the major building blocks of the body tissues, and they are also used as a fuel reserve. There are many different types of foods which are sources of proteins, like poultry, dairy products, fish, eggs, milk, all these things. They are all sources of proteins. In the human body, whenever we take in any food containing protein, the proteins are broken down in the stomach into smaller chains by the hydrochloric acid which are present in the stomach. And this is only possible through the action of certain enzymes. So in this particular topic, we are going to be looking at the chemistry behind the formation of the protein, as well as how proteins can get denatured. Acids come together to make up a large molecule called a protein. There are 9 essential amino acids which humans must obtain from their diet in order to prevent them from protein malnutrition. And apart from them, there are certain amino acids which are synthesized in the human body for the regular functioning of this. Now, the amino acids are actually made of 2 functional groups. So these functional groups are an amine group and a carboxyl group. The amino acid can act as both an acid and a base. And the reason why it is able to do that is because of the functional group that has a central carbon, which is attached to 4 different groups. Sometimes it can be 2 same groups depending on the R group. Now, first group is the amine group, the second group is the carboxyl group, the third group is the hydrogen group, and the fourth one is the R chain. This R can be a hydrogen, a CH3, or any other group. And each of these R group is going to be identified as a different amino acid. So when amino acids join together, they form a dipeptide. When dipeptides join together, they form a polypeptide. When many polypeptides join together, they form a large structure which is known as protein. So this is how the sequence of protein formation takes place. In order for protein to form, we need to assemble the basic building blocks which is called the amino acids. Amino acids join together to form a peptide bond. And in this process, there is a loss of water molecule. Whereas in a hydrolysis reaction, enzymes are used to break a peptide bond and it will form amino acids one more time. The use of water is going to initiate this hydrolysis reaction. Is this. The carboxyl group is going to lose the OH. The amine group is going to lose the H. And together they are going to form water as a byproduct. This is a group that has been brought down after the OH has been lost. The process is going to continue until a longer and a larger chain of protein is created. So in the human body, when digestion takes place, Any protein food that we eat, like meat, poultry, dairy product, anything that contains protein, when we consume that, the enzymes break the protein down into different amino acid. Then these amino acids will assemble themselves one more time to form different protein that is required in different parts of the body. Our body is made of proteins in different places. Like for example, the skin has a different kind of protein. Hair has a different kind of protein. Nails has protein structure as well. Our DNA structure is also a protein. Likewise, we have so many different organs and cells in our body which are protein. Okay, so we have a lot of protein molecules inside our body, long chains of protein structures whose functions are different. Proteins undergo is called hydrolysis reaction. It is this reaction which allows a protein structure to be broken down into various amino acids with the help of enzymes and acids and water to initiate the reaction. Virus substances are proteins in nature, so they carry a protein code, which is why when there is a viral outbreak, it is recommended to use sanitizers or anything that is basic in nature so that the virus can be killed. And the reason why it is possible is because since a virus is a protein structure, the alcohol in the sanitizer is able to break down the peptide bond in the virus. Same thing with a base. Bases like hypochlorite ion. Hypochlorite ion in bleaches are bases, and these bases are able to break the peptide bond in the virus, which allows the virus to denature. So the same thing happens to all the protein structures. Now, if you look at this example, a dipeptide molecule into different amino acids. So let's try to circle and identify one amino acid from this particular chain. So what you have to look for is a carbon atom. Okay, so we are going to be looking at this particular carbon atom. This carbon atom is bonded to 4 different groups. So there is a hydrogen group here, there is an R group here, a nitrogen group, and a carbonyl group. So these are the 4 groups that are attached to this particular amino acid. Similarly, we have another amino acid which is going to be, okay, this is another amino acid, and then there is a continuation on this side. And there is another one which is going to continue on the other side here. So when we are going to identify the peptide bond is formed between this particular carbon here. Alright, that's a carbonyl carbon, the carbon and a double bond O. And the nitrogen group is this one. So this dipeptide bond is going to break now. And once the dipeptide bond is going to be broken, you can see that peptide bond, you can see that the different amino acids are now going to be this particular group as one amino acid and this particular group as another amino acid. So what is missing from group which contains H plus and OH minus? They are going to go and attach themselves respectively to the different molecules. So the OH is going to go and attach itself here, while the H is going to go and attach itself to this nitrogen here. And that is what we see in the two structures. So this is how our peptide bond can be broken down into different amino acid with the action of acids and enzymes. So that's all we have on protein structure. I hope you have learned a lot about carbohydrates, proteins and fats. In the next session of this particular strand, we are going to continue into the different types of chemicals which are used in medicines, in drugs, and also the different kind of additives which are added in the food. Until then, you take care. Bye.